Hi everyone, I am G.DHARANI, Assistant Professor of Biochemistry, A.D.M.COLLEGE for Women, Autonomous, Nagapattinam. Today we will see the topic is cell. Introduction. Fundamental units of life, cells replicating structure. Then next, the first cells were absorbed and named by Robert Hooke in 1665 from slice of cork. Some organisms consist of a unicellular organism, others are multicellular. The cell diameter is 2 to 120 μm. Okay. The cell contains 12 organisms: mitochondria, Golgi complex, ribosome, vacuole, centriole, cell membrane, cytoplasm, lysosome, endoplasmic reticulum, DNA, and final nucleus. Okay. Mitochondria. Mitochondria are membrane-bound cell organelles that generate most of the chemical energy needed to power the cell's biochemical reaction. The chemical energy produced by the mitochondria is stored in the small molecules called adenylyl triphosphate. This is called as ATP. Then Golgi complex. A Golgi body, also known as Golgi apparatus, is a cell organelle that helps process and package protein and lipid molecules, especially protein designed to be exported from the cell. Named after this discovered Emilio Golgi, the Golgi body appears as a series of stacked membranes. Then next, ribosome. A ribosome is an intercellular structure made of both RNA and protein, and it is the slide of protein synthesized in the cell. The ribosome reads the messenger RNA. This is called as mRNA. Synthesize and translate that genetic code into a specified string of amino acids which grow into a long chain that fold to form protein. Then endoplasmic reticulum. The endoplasmic reticulum is otherwise called as ER. It contains two types: rough endoplasmic reticulum, then other one is smooth endoplasmic reticulum. Okay, the endoplasmic reticulum is a large dynamic structure that serves many roles in the cell, including calcium storage, protein synthesis, and lipid metabolism. The endoplasmic reticulum reverse function of the ER are performed by dendritic domain consisting of tubules, sheets, and the nuclear envelope. Okay. Finally, nucleus. A nucleus, as related to genomics, is the membrane enclosed organelle within a cell that contains the chromosome. An array of fold-up pores in the nuclear membrane allow for the selective passage of certain molecules such as protein and nucleic acid into and out of the nucleus. Human cell 3D. Okay, this is the theory of the cell theory produced in Statue Chavan in 1839. Okay, all living things are made up of cells. The next, cells are the smallest working unit of all living things. The next, all cells come from pre-existing cells through cell division. Okay, then different substances that make a cell are connectively called protoplasm. The protoplasm is composed of water, carbohydrate, lipid, protein, and finally electrolyte. Water. 70 to 80% of water is present in the cell. Electrolyte. Sodium, potassium, magnesium, calcium, phosphate, chloride, and finally bicarbonate. Okay. Major substances present in the cells are three types. One is cell membrane, then second one is cytoplasm and its organelle, then finally nucleus. Cell membrane, it is the first. This is thin fibrillable elastic outermost structure which envelope the cell. It consists of bilipid layer with enveloped protein that are integral protein, then peripheral protein. Okay, this is the function of the cell membrane: productive, digestive, selective. Variability is contained two types: non-polar molecules and polar molecules. Okay, productive. From outermost boundary of the cell organelles. Digestive takes in food and excretes waste products. Collectively, then it contains two types, non-polar and polar. Non-polar gases like O2, oxygen, carbon dioxide, nitrogen. Lipid, steroid hormone, alcohol can dissolve in the non-polar region of the membrane and move rapidly across the membrane. Polar molecules, H2O. H2O is called as water. Water soluble ions, glucose, urea, etc. have much lower solubility. Therefore, penetrate the membrane much more slowly. Then, chemical and physical properties of membrane control the free package of ions in and out of the cell. This properly help in maintaining component in ICF and ECF. Okay. This next last point in soluting properties. It acts dielectric material of a charged conductor. The cell membrane have very high insulating value. Okay. This is the cytoplasm and its organelles. Cytoplasm. It is thick, gel-like, semi-transparent fluid that is found in both plants and animal cells. These constituent part of the cytoplasm are cytochrome, cell organelle, cytoplasmic infusion, bounded by the plasma membrane and contain many organelles in a eukaryotic cell. Cytosol. These cytosol, the aquatic parts of the cytoplasm outside of the organelle also contain its own digestive protein. It accounts for almost 70% of the total cell volume. Generative substance containing mainly of cytosilicon filaments, organic molecules, salt and water. Chemically, the cytoplasmic matrix is composed of many chemical elements in the form of atoms, ion and molecules. This is the organelles. Following organelles are present in the cytoplasm. Mitochondria, ER means endoplasmic reticulum, lysosome, Golgi apparatus, peroxisome, vacuoles. Each organelle is bounded by a lipid membrane and has specific function. First of all, we call mitochondria. This mitochondria was first observed by Follicular in 1815 as granular structure in the vacuoles. Mitochondria are otherwise called as powerhouse of the cell structure. This length is 5 to 12 μm, then diameter is 0.5 to 1 μm. Filamentals are globular in shape. Components of mitochondria are outer membrane, inner membrane. Then intermediate space between outer and inner membrane, cystine, enfolding in intermembrane matrix. The space enclosed by intermembrane. The membranes are made up of phospholipids and protein. Outermost membrane. It contains large number of integral membrane protein called proteins. These proteins from canal that allow molecules of 500 atoms and less to pass. Then intact. It is otherwise called a matrix of mitochondria. Contains enzymes concurred with citric acid cycle and respiratory chain oxidation. Major metabolic pathway involved in oxidation of carbohydrate, lipids and amino acids, and part of special biosynthetic pathways involving urea and heme synthesis are located in the matrix. Okay, then inner membrane. It contains ATPs and other enzymes concerned with the synthesis and metabolism of ATP. Contains enzyme of electron transport chain. The ultimate process of this mechanism is oxidative phosphorylation and the synthesis of ATP. Mitochondria has some protein synthesis by mitochondrial DNA. Then this function, mitochondria function, power generating units of the cell. The next, important to maintain proper concentration of calcium ion within the various compartments of the cell. Energy transcription, growth, respiration, then endoplasmic reticulum. Otherwise called as ER. Okay, network of tubular and fat vesicular structure in the cytoplasm. An extensive network of closed plantar membrane bounded sets called cisternae. Space inside the tubules is filled with endoplasmic matrix. Okay, it contains two types, smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth endoplasmic reticulum. Ribosome is absent. Site of synthesis of lipid and steroid hormone, mainly present in lipid forming cells such as adipocytes, intercell cells of testes, glycogen stored cells of liver, antennal cortex cells, muscle cells, leukocytes, etc. Then rough endoplasmic reticulum contain ribosome, site of protein synthesis, processing and packaging, mainly present in protein forming cells such as pancreatic cells, globule cells, antibody producing plasma cells, and of nerve cells, etc. Okay, this is the function of the endoplasmic reticulum. Synthesis of protein, protein C generation, unsaturation of fatty acids, muscle construction. Here is commonly known as sarcoplasmic reticulum is muscle fiber. This is the Golgi bodies. Golgi body is a collection of membrane enclosed sac composed of 4 or more stacked layer of thin, flat, enclosed vacuous linear cell of the nucleus. Consists of multiple discrete compartments. Consists of 4 functionally discrete regions. One, this is Golgi network, then other one is Golgi stack, then finally transub, then finally transub Golgi network. Okay, this is the function of the Golgi complex. Wrapping and packaging department of the cell, then produces secretion granules, for example, membrane enclosed complex, which stores a hormone and enzyme in the protein secreting cell, is packaging protein. Site of the formation of the lysosome, for example, large irregular substance surrounded by the membrane which are present in the cytoplasm. It adds certain carbohydrates from glycoprotein which play the important roles in the association of the cell from tissue. This is the lysosome. This is one of the cell organelle. Its diameter is 250 to 750 nanometer. They are the irregular structures surrounded by the unit membrane, more acidic than results of the cytoplasm, and external bacteria as well as own out cell components are digested in them. Then integral is kept acidic by the action proton pump or electron or ATPase. Lysosomes are cell hydrolysis and their function based at the acidic pH. Okay, this is the function of the lysosome. Act as the drum of digestive system of the cell because the enzyme present can digest especially all macromolecules. Engulf form out component of the cell in which they are located. Engulf exogenous substance, for example, by bacteria, and degenerate them. When a cell dies, lysosomal enzyme causes autolysis of the rearrangement. That way, lysosomes are also called a suicide batch. Okay, this is the peroxisome. This is one of the cell organelle. Peroxisome diameter is 0.5 μm. A lipid bilayer membrane surround which generate with enter of the peroxisome. Alterate oxidase cisternate cross. Structure is similar to that of the lysosome but with a different composition. Peroxisome can be formed by the building of ER or by division. Okay. Contains oxidase that produce H2O2. Catalyze regenerate hydrogen peroxidase to yield water and oxygen. Proteins are directed to the peroxisome by a unique signal sequence with the help of protein peroxines. This is the function. H2O2 metabolism and detoxification help in photorespiration in plants, biosynthesis of lipids, cholesterol and alcohol are synthesized in animals, synthesis of bile acid in liver, synthesis of plasmalogens. Okay, this is the cytoskeleton. Systems of fiber that not only maintain the structure of the cell but also permit to the charge shape and move. The cytoskeleton made up of primary of macrotubules, intermediate filament, macrofilament along with the protein with anchor tie them together. Microtubules, intermediate filament, macrofilament, microtubules. These are along low structure approximately 25 nanometer in diameter. Determine shape of the cell, role in the construction of the sling and movement of chromosome and centriole, as well as insulin and flagellate motion. Intermediate filament. There are 8 to 14 nanometer in diameter and are made up of various subunits. In their absence, cell rupture more easily and when the abnormal in human in common. The proteins that made up of intermediate filament are cell type specific and the frequency used to cellular market. Macrofilaments. There are long solid fiber 4 to 6 nanometer in diameter. They compose the contractive protein acting and the responsible for the cell motion. This is the function. They are involved in the movement of the chromosome, cell movement, process that move secretion granule in the cell, movement of protein within the cell membrane. Okay. Finally, we see the nucleus. The nucleus. The nucleus contains chromatin, RNA, and nuclear protein move freely in aqueous solution. Nucleus has an internal structure that organizes the genetic material and locates the nucleus function. A loosely organized matrix of nuclear lamins extend from the nuclear lamina into the interior of the nucleus. These lamins serve as site of chromatin attachment and organize other protein into functional nuclear bodies. Chromatin within the nucleus is organized into large loop of DNA and specific region of those loops are bound into the lamin matrix by lamin binding protein in the chromatin. This is the nuclear envelope. Complex structure is the nuclear membrane as underlying nuclear lamella and nuclear pore complex. Two concentric membrane called the inner and outer nuclear membrane. The outer membrane is continuous with the endoplasmic reticulum. So the space between inner and outer nuclear membrane is directly connected within the lumen of the endoplasmic reticulum. The nuclear membrane is permeable only to small non-polar molecules. Underlying the inner nuclear membrane is the nuclear lamina, a fiber meshwork that provides frictional support to the nucleus. This is the function of the nucleus. Okay. So, both are the responsibility of genetic information as a cell control center. The presence of the nucleus those allow gene expression to be regulated by the transcription mechanism such as alternative spelling. The nucleus envelope provides novel opportunities for the control of the gene expression at the level of transcription. Okay, this is the final slide. Summary, component and major function. First we see the components. Plasma membrane, cytosol, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosome, peroxisome, then nucleus. Major function. Transport of ion and molecules, metabolism of carbohydrate, lipid and amino acid, then mitochondria, energy production, ER, synthesis of protein and lipids, Golgi apparatus, modification, spotting of protein, lysosome, cellular digestion, peroxisome, utilization of H2O2, cytosol, cell morphy and cell mobility, nucleus, DNA synthesis and repair. Thank you.